I'm going to go over the Unit 6 study guide with you, and I'm going to break it into the 4 different pages that we have, so that way this isn't a really long video. So first things first, page 1 of the Unit 6 study guide lets you know that the exam is going to be 100 points. 35 of them will be multiple choice. There'll be 5 short answers, and there's just a reminder to make sure that you take your time, read through each question very carefully, and use process of elimination to come up with the best answer. The first question on the study guide asks you to review the 3 Mendel's laws of inheritance. So Gregor Mendel, as we know, is the father of genetics, and he came up with 3 basic laws. The first one is the law of dominance and recessiveness, and what this means is that a dominant trait will always be seen in the phenotype of the offspring, even if there is a recessive allele present. So in the example that I gave on the study guide here, AA, capital, and AA, capital lowercase, you will still see a dominant phenotype expressed. In order for a recessive characteristic to be seen in the phenotype of the offspring, both alleles would have to be recessive, so little a, little a. The second of Mendel's laws is the law of independent assortment, and this refers to the way allele pairs separate, and sometimes you see the word segregate in place of the word separate. It's the way allele pairs separate into 2 daughter cells that has no effect on the way any other allele pairs are separated. So they separate independently from each other. They're not, you can have someone with blue eyes and blonde hair. You could also have someone with blue eyes and brown hair. The alleles separate independently from each other. The law of segregation, and remember, the word segregate means to separate. The law of segregation means that you're going to be getting half of the information from a parent. So a parent may have 2 alleles for a certain gene, each on 1 copy of a chromosome. In the law of segregation, we learn that these 2 alleles will be separated from each other during meiosis. Specifically in the second phase of meiosis, the 2 chromosomes are going to separate from each other, causing 2 distinct alleles located on those chromosomes to separate from each other. So if mom's diploid cell has a big A, little a, if she's heterozygous for that allele, she's going to go through meiosis 1 and then meiosis 2, and so the haploid cells are going to have 1 big A, 1 little a, 1 big A, 1 little a. So she's going to have those alleles separate from each other. Number 2 on the study guide, it says to define heterozygous and homozygous and then to give 2 examples. And I think we really went over that a lot in this unit. So heterozygous is also known as hybrid, also known as being a carrier, and an example is a big H, little h. Homozygous is also known as purebred, so you could be big H, little h, little h. As long as you have 2 of the same letters, you're homozygous. Number 3 should have been really easy for you then. So number 3, homozygous dominant, big H, heterozygous dominant, big H, little h, homozygous recessive, little h, little h. A phenotype refers to a physical characteristic. So the characteristic that I used was brown hair. And number 5 was a really easy cross because you're crossing 2 homozygous dominant parents, big H, showing the probabilities of their offspring. They would all be big H, big H. If you have any questions about page 1, be sure to let me know before the test.